The overall goal of the following experiment is to use a measure the effect of potential regulators of TGF-β-induced invasion in a three-dimensional environment. This is achieved by first making spheroids of the cells. The spheroids are then embedded in collagen, which provides a 3D environment. Next, the invasive area is quantified in order to determine the effect of treatment on TGF-β-induced invasion. The results show that modulators of the TGF-β pathway affect the invasive property of the spheroids in a 3D collagen matrix.
The main advantage of this technique over existing methods, like the scratch assay, is that it better recapitulates the 3D environment.
This method can help to answer key questions in the TGF-β field, such as the identification of novel genes involved in TGF-β-induced invasion.
To prepare methocell solution for the TGF-β-induced invasion assay, begin by autoclaving 6 grams of methylcellulose in a 500 milliliter bottle containing a magnetic stirrer. Dissolve the methylcellulose in 250 milliliters of preheated DMEM F12 without serum for 20 minutes. Add 250 milliliters of room temperature DMEM F12 containing 10% horse serum. Mix overnight at 4 degrees Celsius. The next day, clear the solution by centrifugation at 5000 G for 2 hours at 4 degrees Celsius. Transfer the clear, highly viscous solution to a new tube. Store at 4 degrees Celsius until use. To culture spheroids,